Hello everyone, my name is Francis Fadricela and I will introduce to you the agricultural biotechnology and one of its applications, the pest-resistant plants. For many years, humans have been searching for a way to grow their crops in spite of insect pests. In the past, Asian farmers select pest-resistant genes in their crops, sometimes by action as simple as collecting seed from only the highest-yielding plants in their field. With the beginning of genetic engineering, it became a major benefit to the field of agriculture. Through agricultural biotechnology, scientists were able to make or modify products, improve plants, or even develop microorganisms for specific agricultural uses. Genes for insect resistance now can be moved into plants more quickly and deliberately. BT or Bacillus thuringiensis technology is only one example of ways genetic engineering may be used to develop insect-resistant crops now and in the future. So, what is Bacillus thuringiensis? Bacillus thuringiensis, or BT, is a species of bacteria that lives in soil. This bacteria can produce proteins that are toxic to certain insects when ingested. It may be toxic to insects, but these bacteria have little or no effect on other creatures that consume it, like humans or other mammals. BT was first discovered in 1901 by Japanese biologist Shigeru Ishiwatari, and it was rediscovered in 1911 by Ernst Berliner. He named it Bacillus thuringiensis after a German town, Thuringia, where the moth was found. Ishiwatari had named the bacterium Bacillus sotum in 1901, but the name was later ruled invalid. In 1928, BT was used as an insecticide spray for pest control. BT was first available as a commercial insecticide in France in 1938, and in 1950s it entered commercially in USA. The use of BT in 1980s increased when insects became increasingly resistant to synthetic insecticides. The scientists and the environmentalists became aware that the chemicals were harming the environment. BT is organic and it affects specific insects and does not persist in the environment. Because of this, government and private industries started to fund research on BT. Further studies and experiments were done, and in 1995, the first genetic engineered corn was made with the genes of BT. Today, there are thousands of strains of BT. Many of them have genes that encode unique toxic crystals in their DNA. There are many different strains of BT with different proteins specifically lethal to certain insects, as you can see in the table. This is how BT plants are engineered to produce their own BT toxins. This is done by copying certain pieces of DNA from BT and putting them into a plant's DNA. Plants use this DNA to produce BT toxins within its own tissues. In that way, when insects eat the plant's tissues, they would be exposed to the toxins. The BT genes were used in corns, cottons, eggplants, potatoes, and many more. They are called BT crops, which are highly effective to combat pest problems. BT crops have advantages and disadvantages. The advantages are BT crops are non-toxic to humans and other animals. And because of its pest resistance, it improves the crop's quality and also reduces the use of synthetic pesticide that can cause soil pollution. It would also raise farmers' incomes and beneficial to the farm's production. It would also be more productive in a small area of land. For the disadvantages, it can disrupt the natural process of gene flow. And because of its genetically modified, it would cost more budget than natural grown crops. And over time, insects became more resistant to toxins. That's all, and thank you.